Welcome everyone. Today our group will present about one of 6 hallmarks of cancer, which is the tissue invasion and metastasis.
Cancer cell from primary tumor, which is on the left on the picture, move through endothelial cell of blood vessel wall to enter the bloodstreams, in which they will move to another organ of the body. Then they move out of the blood vessel by passing through endothelial cell one more time to form other distant metastasis tumor at that organ.
As you know, tissue invasion and metastasis is the ability to escape primary tumor mass and colonize new terrain in other parts of tissue and other organs. The first features of this hallmark is the loss of E-cadherin. E-cadherin is one of the factors that contribute to the mechanism of contact inhibition, which means cells that touch one another do not grow any further. Unsurprisingly, E-cadherin function is lost in migrating cancer cells. Conversely, another molecule known as N-cadherin shows increased level in migrating cancer cells. As this molecule helps the cancer cell to slip through blood vessels during migration. Overall, E-cadherin expression is stopped so that the segment that binds the cancer cells to the other cells is eliminated. The expression of N-cadherin is enhanced so they can move through blood vessels to distant sites more efficiently. Another trait which I would like to talk about is the untethering of bonds between cell and extracellular matrix, or ECM. Our tissues are not made up solely of cells. A large proportion of tissue consists of extracellular space, which is filled with a mixture of carbohydrate and protein molecules. This space is known as the ECM. The molecules that make up the ECM are secreted by cells embedded in it, and these cells tether themselves to the ECM to form the tissue. Also, metastasis therefore requires the untethering of this bond to allow predaceous cancer cells to migrate freely.
Normal cells grow and divide but have many control on that growth. On the other hand, cancer cells have defects in the control mechanism that govern how often they divide, and the feedback systems that regulate this control mechanism. There are 6 signaling pathways: cell sufficiency in growth signal, insensitivity to anti-growth signal, tissue invasion and metastasis, limits replicative potential, such as angiogenesis and evading apoptosis. Until 2010, the scientists update more 4 signals. That is, they regulate metabolism, evading the immune system, unstable DNA, and inflammation. Nowadays, by applying a system biology approach to the cancer hallmark network framework, hallmark network can be quantified and then computationally modeled to predict cancer and evolutionary path and clinical phenotype, which is beyond the text-only description of the cancer hallmark. Now I will talk particularly signals. This is tissue invasion and metastasis. Cancer cells can break away from their site or organ of origin to invade surrounding tissues and spread to distant body parts. Primary tumor mass beyond cells that invade other tissues and may then travel to distant sites and establish metastasis. The newly formed metastases as amalgam of cancer cells and normal supporting cells consecrated from the host tissues. Metastatic cells must mimic normal cell-to-cell interaction through cell-self interaction, cell-self adhesion molecule as known as CAM and integrin. NCAM is normal adhesion suppressing metastasis, but it becomes less and allow metastasis in grim tumor, neuroblastoma and small cell lung cancer, and it spread reduce its invest pancreatic and colorectal cancer. Ecaherin, which is spread to the epithelial cells, transmit anti-growth signal. Ecaherin is therefore a widely acting suppressor of invasion and metastasis by epithelial cells, which must be overcome by cancer cells to progress. Integrin display subjects, friend neurons and genes in the integrin are displayed by migrating cells. Matrix degrading proteins are also necessary to facilitate invasion into stroma across the blood vessel wall and through normal epithelial cell layer.
Now I will present to you the diagnosis of tissue invasion and metastasis. There are several strategies to diagnose the metastasis. We can use the X-ray to scan for the lung, liver, abdominal, or the pelvis metastasis. We can also use the bone scan, the computerized tomography, also known as CT, the magnetic resonance imaging, and the blood test, and finally the positron emission tomography. The magnetic resonance imaging is a medical imaging technique used in radiology to investigate the anatomy and physiology of the body. This technique is used to diagnose the stage of disease, and the blood tests can be applied to identify the liver and bone metastasis, and the positron emission tomography is used to scan the metastasis throughout the body. It produces a three-dimensional image of functional processes in the body. One of the common strategies in identify the tissue invasion and the metastasis is immunohistochemistry. This is a process of detecting antigens, for example, the proteins in cells of a tissue section, by exploiting the principle of antibodies binding specifically to antigens in biological tissues. Immunohistochemistry staining is widely used in the diagnosis of abdominal cells, such as those found in cancerous tumors. Specific molecular markers are characteristic of particular cellular events, such as the cell proliferation or cell apoptosis. The process of immunohistochemistry is carried out with two major steps. First is the sample preparation. It involves tissue collection, fixation, and sectioning. This step is aimed to maintain the cell morphology and the tissue architectures. Sometimes it requires to block or quench endogenous biotin or enzymes to maintain the antigenicity of target epitopes. This is aimed for the antibody staining. The second step is sample labeling. We can use antibodies, the monoclonal ones or the polyclonal. To target the antigen, we can apply the direct method, which is a one-step staining method involving a labeled antibody reacting directly with the antigen in tissue sections. Or we can apply the indirect method, which is an unlabeled primary antibody that binds to the target antigens in the tissue, and a labeled secondary antibody that reacts with the primary antibody. In the immunohistochemistry, reporters are used based on the nature of detection method, and the most popular reporters are the chromogenic and fluorescent detection, which are mediated by an enzyme or a fluorophore. To identify the tissue invasion and metastasis, we use the immunohistochemistry markers. For example, the cytokeratins are used to identify carcinomas. The alpha-fetoproteins are used to identify the hepatocellular carcinoma. To target the gastrointestinal stromal tumor, we use the CD117 and the CD10 for renal cell carcinoma and acute lymphoblastic leukemia. An example of immunohistochemistry method is the evaluation of central nervous system metastasis with the immunohistochemistry correlation. This is a study of 74 patients with unknown metastasis. And in this study, we use plenty of antibodies, for example, the keratinin, the cytokeratin 20, cytokeratin 7, the chromogranin, and so on. And after the immunohistochemistry, we can obtain the result as the metastases are identified. In this example, 51 persons have lung metastasis, 10 patients are not diagnosed which metastasis they have, and 4 have the female genital tract metastasis, and so on. Another strategies for diagnosis of metastasis is the Oncotype DX. Oncotype DX is a test that helps predict the chance of metastasis when cancer spreads to other organs. For some breast cancer, it tests the sample of the tumor, which is removed during a biopsy or surgery for a group of 21 genes. This test provides more information about how likely or unlikely the breast cancer is to come back, and it also predicts the likelihood of benefit from chemotherapy or radiation therapy treatment. Along with other prognosis and diagnosis factors, the results of Oncotype DX tests can be used to tailor treatment plans. We obtain the result from the Oncotype DX as a number between 0 and 100. It is called a recurrence score. If the recurrence score is smaller than 18, the cancer has a low risk of recurrence. And the benefit of chemotherapy for early stage breast cancer or the radiation therapy is likely to be small and will not outweigh the risk of side effects. And if the recurrence score is larger than 18 and smaller than 31, the cancer has an intermediate risk of recurrence. It is unclear whether the benefits of chemotherapy for early stage breast cancer or radiation therapy outweigh the risk of side effects. And finally, if the recurrence score exceeds 30, the cancer has a high risk of recurrence, and the benefits of chemotherapy for early stage breast cancer or radiation therapy are likely to be greater than the risk of the side effects. In this way, Oncotype DX may help some people avoid chemotherapy and its side effects. Oncotype DX helps predict the chance of metastasis for breast cancer that are all of the following: stage 1 or 2, estrogen receptor positive, or HER2 neu negative, or lymph node negative. Researchers are studying whether Oncotype DX can predict how well breast cancer will respond to different chemotherapy drugs. The Oncotype DX test for invasive breast cancer has been modified to help predict the chance that ductal carcinoma in situ, in short for DCIS, will return as DCIS or invasive breast cancer. This could help identify which cases of DCIS would benefit most from radiation therapy after lupectomy. This test needs further study and is not yet part of standard practice.
Now we will talk about cancer cell biomarker in tissue invasion and metastasis. Cancer biomarkers are substances indicate that tumor stage, progression, characteristic, and response to therapy. Most cancer biomarkers are transcription factor, cell surface receptor, or secrete protein that are produced by either cancer cell or other cell in response to cancer. They are present in tumor tissue or body fluid. Cancer biomarker can be used for screening the general population for differential diagnosis and for clinical staging of cancer. Additionally, cancer biomarker can be used to estimate tumor volume, to evaluate response to treatment, to assess disease recurrence through monitoring, or as a Additionally, cancer biomarker can be used to estimate tumor volume, to evaluate response to treatment, to assess disease recurrence through monitoring, or as a prognosis indicator of disease progression. Using the lab decade improved understanding of carcinogenesis and tumor progression has revealed a large number of potential cancer markers. It is predicted that even more will be discovered in the near future will be application of the new emerging genomic and proteomic technology. Each type of cancer have a different biomarker. So here we just mentioned about biomarker of breast cancer. Firstly, we will talk about the estrogen receptor, progesterone receptor, and SER2. The biomarker are only three validated validated biomarker routine apply in breast cancer management. ER, PCR, and SER2 expression provide prognosis information and inform on decision regarding therapy. One of the hallmarks of cancer is genomic instability, indicating that genetic mutation and hence alteration in both downstream pathway signaling and protein expression are possible if not expected. The expression of hormone receptor ER and PR in a patient's breast cancer is an example of a weak prognosis but strong predictive biomarker. If a patient's tumor expresses ER and or PR, as seen in approximately 70% of invasive breast cancer, we can predict that this patient will likely benefit from endocrine therapy such as tamoxifen. The overexpression of the oncogenes SER2 in a patient's breast cancer, as seen in approximately 15 to 20% of invasive breast cancer, is an example of both prognosis and predictive biomarker. SER2 expression is associated with diminished prognosis. However, it also predicts that a patient will more likely benefit from anthracycline and taxane-based chemotherapy and direct therapy that target SER2 but not to endocrine-based therapy. Disorder between primary tumor and MCB tissue occurred at rates of around 10% to 30% for ER and 20% to 50% for PR, with glow being more common than gain. SER2 discordance has been reported in around 10% of primary and metastasis tumor, with loss and gain occurring with relatively equal frequency. While there is a reasonable rationale for the development of receptor, and the vast majority of data have been derived retrospective, creating some uncertainty about the true disorder rates that may exist between primary and current breast cancer. Another biomarker of breast cancer is miRNAs. MicroRNAs are small non-coding RNA molecules involved in both transcriptional gene regulation and have recently been shown to play a role in cancer metastasis. In solid tumor, especially breast cancer, alternation in miRNA expression contribute to cancer pathogenesis, including metastasis. Considering the emergence of miRNAs in metastasis, the identification of predictive marker is necessary to further the understanding of stage-specific breast cancer development. This is a retrospective analysis that aims to identify molecular biomarkers related to distant breast cancer metastasis. 7 microRNA are identified in metastasis group in respective of clinical staging. SSA-7A, SSA-7B, SSA-7C, SSA-MIR1308, SSA-MIR21, SSA-MIR494, and SSA-MIR923. Of these, SSAMIR494 and SSAMIR21 were selected for further analysis using real-time PCR. As you can see in the picture, the amount of this microRNA in the metastasis tumor is very high compared with the non-metastasis tumor. That's why this microRNA was used as a biomarker in tissue invasion and metastasis. Another biomarker in breast cancer is DP103. We define DP103 as a biomarker and metastasis-driving oncogene in human breast cancer and determine that DP103 is DP103 elevate basic metallopeptidase 9 MMP9 level, which are associated with metastasis and invasion through activation of NF-κB intern NF-κB signaling property. Active DP103 expression. Furthermore, DP103 enhances TGF-beta activated kinase. Furthermore, DP103 enhances TGF-beta activated kinase 1, phosphorylation of NF-κB activating IKB kinase 2, leading to increased NF-κB activity. Reduction of DP103 expression in invasive breast cancer cells reduce phosphorylation of IKK2, abrogate NF-κB mediated MMP9 expression and impeded metastasis in a murine xenograft. In breast cancer patient tissue, elevated level of DP103 correct. In breast cancer patient tissue, elevated level of DP103 correlated with enhanced MP9, reduced overall survival and reduced survival after relapse. Together, the data indicates that a positive DP103 NF-κB feedback loop promotes constitutive NF-κB activation in invasive breast cancer, and activation of this pathway is linked to cancer progression and the acquisition of chemotherapy resistance. Furthermore, our results suggest that DP103 has potential as a therapeutic target for breast cancer treatment. Now we will move on to drug targets of tissue invasion and metastasis of cancer cells. In this case, we just mentioned about the heat shock protein 90. The heat shock protein 90, or we call it as SSP90, belong to the family of molecular chaperone responsible for the conformational maturation or reparation of other protein, referred to as client into biologically active structure. SSP19 exists in its essential ATP-dependent chaperone function to more than 300 client proteins involved in the cell growth, differentiation, and survival. Many of them, more than 40, include overexpression or mutant oncogenic protein, transcription factor, hormone steroid receptor, and ER or AR, and angiogenic factor SIF1 alpha, which are associated with the 6 hallmark of cancer. SSP90 is a molecule type 1 required for the stability and function of numerous oncogenic signaling proteins that determine the hallmark of cancer: receptor, tyrosine kinase, and hypoxia-inducive factor. Medicated pathway commonly activates in aggressive cancer, potentiates each other, and thus efficiently promote cancer invasion and metastasis. SSP19 inhibitor, by interacting specifically with a single molecule, SSP19 causes the destabilization and eventual degradation of multiple SSP19 clean protein. The agent impedes the cellular process involved in cancer invasion and metastasis by simultaneously impairing multiple SSP19 dependent signaling protein. SSP90 play a central role in supporting all of 6 hallmark traits of cancer suggested by Hanahan and Weber by chaperoning a subset of clean protein as shown. SIF1 alpha, hypoxia-inducible factor 1 alpha, VEGF, vascular endothelial growth factor, and IKK inhibitor of kappa B kinase.
Next part, I will tell you about the recent remarkable findings. One of the findings very popular is in the August 15, 2009 issue of Gene and Development, Liu and colleagues, page 1882 and 1894, propose a novel molecular mechanism by which tumor produce metalloproteinase release epidermal growth factor, ligand to an active center of osteoclastogenic pathway, receptor activator of NFKB ligand to promote the breast cancer osteolysis. The next finding is about a new publication in Cells by 2013 uncover a subpopulation of cells in luminal breast tumor with characteristic of basal epithelium that lead a multicellular collective invasion of surrounding tissue. The process of collective invasion, as uncovered by this author, occurs through cell-cell interaction among functionally and behaviorally distinct cells with unique molecular signature. The therapeutic targeting of these cells within the primary tumor may help to prevent metastasis of a wide range of aggressive cancer. The next finding is about the device monitor key step in development of tumor metastasis in Science, 18 August 2014. A microfluidic device may help study key steps in the process by which cancer cells break off from a primary tumor to invade other tissue and form the metastasis. This device gives us a platform to be used in testing and comparing compounds to block or delay the epithelial mesenchymal transition, potentially slowing the progression of cancer. Another finding is about the small GTP ASA RAB37 targets tissue inhibitor of metalloproteinase 1 for exocytosis and thus suppresses tumor metastasis, published in 3rd September 2014. RAF small GTPase are master regulator of membrane trafficking and guide vesicle targeting. Recent publications show that RAF control the trafficking pathway are entered during the tumorigenesis. Next finding is about the integrated genomics epigenomic analysis of breast cancer brain metastasis. It was published in January 12, 2014. They highlighted a number of fundamental genetic and epigenetic aberrations occurring in brain metastasis from primary breast tumor with strong implications for the future targeted therapy that are aimed at alleviating the burden of this clinical unmet need.
Next, I would like to present about some major gaps of knowledge in invasion and metastasis. The mechanisms remain poorly understood and methods in combating metastasis remain limited. There is too little knowledge of how the many events are coordinated and integrated by the cancer cell with conspiratorial head by the stromal component of the host. Therefore, before drug development can proceed with a legitimate chance of success, significant gaps in basic knowledge need to be filled in order to discover promising new therapeutic approaches targeting the microenvironment. Secondly, the ability to invade and metastasize distant sites is a key hallmark of cancer progression and possibly one of the most complicated and least understood hallmarks so far. Metastasis has been traditionally thought of as the final stages of a burgeoning tumor. Indeed, our current classification system for a tumor upon diagnosis assigns a higher value when there is evidence of metastasis. However, recent evidence suggests that metastasis does not necessarily happen in the final stages of cancer progression, but can occur at any time, sometimes even before the primary tumor is observed. Thirdly, fundamental gaps exist in our knowledge of how malignant cells overcome the cytostatic actions of TGF-beta and of how TGF-beta stimulates the acquisition of cancer hallmarks by developing and progressing human cancers. Tumorigenesis counteracts the tumor suppressing activities of TGF-beta, thus enabling TGF-beta to stimulate cancer invasion and metastasis. And finally, once a cancer has metastasized, it may still be treated with radiosurgery, chemotherapy, radiation therapy, biological therapy, hormone therapy, surgery, and or a combination of these interventions. The choice of treatment depends on a large number of factors, including the type of primary cancer, the size and location of the metastasis, the patient's age and general health, and the types of treatments used previously, among others. The major gap of knowledge here is to choose a suitable treatment because the treatment options currently available are rarely able to cure metastatic cancer, though some tumors such as testicular cancer and thyroid cancer are usually still curable.
Now we will talk about the future research and therapy of
tissue invasion and metastasis. First, I will talk about the studying mechanism of upon protein inhibitor treatment. Proteae are involved in normal destructive event and tissue remodeling. The most likely mechanism by which proteae could mediate metastasis is by catalyzing degradation of extracellular matrix and basement membrane. It concludes that if proteae could be provoked to play a role in the invasion of cancer, inhibition of the enzyme could open up new therapeutic approaches for the control of malignancy. Besides studying the role of proteae, especially on urokinase type, plasminogen activator, and collagenase 4, we also suggest that both adhesion protein and motility factor could also serve as target for new treatment to prevent cancer invasion and metastasis. Therefore, some of the inhibitors of experimental metastasis which are described will be evaluated in clinical trial in the near future. The first one is we can test the diagnostic accuracy of detection tissue invasion in patients. We suggest that the number of patients at each stage of the disease be specific, with the aim of discovering the proportion of patients classifiable clinically as with invasion and without invasion. In the same way, it is necessary to clarify whether patients who have received previous treatment should be included, because in previously irradiated patients, invasion tend to be more extensive and less predictable. The final research is blocking the interaction between glycosaminoglycan and cancer cell protein. The glycosaminoglycan chondroitin sulfate type E consists of repeating units of disulfide and acetylgalactosamine linked to glucuronic acid, and is known to bind to the cell motility regulators 16 and CD44. Using a new CSE-specific antibody, detected CSE in various mouse models of spontaneous pancreatic cancer. Cleavage of the extracellular domain of CD44 by metalloproteinase is induced by hyaluronate, where the CSE is generated by tumor metalloproteinase or other unknown mammalian chondroitinase remain to be determined.